15.4 RNA processing in eukaryotes. By the end of this section, you will be able to do the following. Describe the different steps in RNA processing. Understand the significance of exons, introns, and splicing for mRNAs. Explain how tRNAs and rRNAs are processed after transcription. Eukaryotic pre-mRNAs must undergo several processing steps before they can be translated. Eukaryotic and prokaryotic tRNAs and rRNAs also undergo processing before they can function as components in the protein synthesis machinery. mRNA processing. The eukaryotic pre-mRNA undergoes extensive processing before it is ready to be translated. Eukaryotic protein coding sequences are not continuous, as they are in prokaryotes. The coding sequences, exons, are interrupted by non-coding introns, which must be removed to make a translatable mRNA. The additional steps involved in eukaryotic mRNA maturation also create a molecule with a much longer half-life than a prokaryotic mRNA. Eukaryotic mRNAs last for several hours, whereas the typical E. coli mRNA lasts no more than 5 seconds. Pre-mRNAs are first coded in RNA-stabilizing proteins. These protect the pre-mRNA from degradation while it is processed and exported out of the nucleus. The three most important steps of pre-mRNA processing are the addition of stabilizing and signaling factors at the 5' and 3' ends of the molecule, and the removal of the introns, figure 15.11. In rare cases, the mRNA transcript can be edited after it is transcribed. Figure 15.11. Eukaryotic mRNA contains introns that must be spliced out. A 5' cap and 3' poly A tail are also added. Evolution connection RNA editing in trypanosomes. The trypanosomes are a group of protozoa that include the pathogen Trypanosoma brucei, which causes nagana in cattle and sleeping sickness in humans throughout great areas of Africa, figure 15.12. The trypanosome is carried by biting flies in the 380 Chapter 15, Genes and Proteins. Access for free at openstax.org. Genus Glossina, commonly called tsetse flies. Trypanosomes, and virtually all other eukaryotes, have organelles called mitochondria that supply the cell with chemical energy. Mitochondria are organelles that express their own DNA and are believed to be the remnants of a symbiotic relationship between a eukaryote and an engulfed prokaryote. The mitochondrial DNA of trypanosomes exhibit an interesting exception to the central dogma. Their pre-mRNAs do not have the correct information to specify a functional protein. Usually, this is because the mRNA is missing several U-nucleotides. The cell performs an additional RNA processing step called RNA editing to remedy this. Figure 15.12. Trypanosoma brucei is the causative agent of sleeping sickness in humans. The mRNAs of this pathogen must be modified by the addition of nucleotides before protein synthesis can occur. Credit. Modification of work by Torsten Oxenreiter. Other genes in the mitochondrial genome encode 40 to 80 nucleotide guide RNAs. One or more of these molecules interacts by complementary base pairing with some of the nucleotides in the pre-mRNA transcript. However, the guide RNA has more nucleotides than the pre-mRNA has U-nucleotides with which to bind. In these regions, the guide RNA loops out. The 3' ends of guide RNAs have a long poly-U tail, and these U bases are inserted in regions of the pre-mRNA transcript at which the guide RNAs are looped. This process is entirely mediated by RNA molecules. That is, guide RNAs, rather than proteins, serve as the catalysts in RNA editing. RNA editing is not just a phenomenon of trypanosomes. In the mitochondria of some plants, almost all pre-mRNAs are edited. RNA editing has also been identified in mammals such as rats, rabbits, and even humans. What could be the evolutionary reason for this additional step in pre-mRNA processing? One possibility is that the mitochondria, being remnants of ancient prokaryotes, have an equally ancient RNA-based method for regulating gene expression. In support of this hypothesis, edits made to pre-mRNAs differ depending on cellular conditions. Although speculative, the process of RNA editing may be a holdover from a primordial time when RNA molecules, instead of proteins, were responsible for catalyzing reactions. 5' capping. While the pre-mRNA is still being synthesized, a 7-methylguanosine cap is added to the 5' end of the growing transcript by a phosphate linkage. This functional group protects the nascent mRNA from degradation. In addition, factors involved in protein synthesis recognize the cap to help initiate translation by ribosomes. 3' poly A tail. Once elongation is complete, the pre-mRNA is cleaved by an endonuclease between an AAUAAA consensus sequence and a Gurik sequence, leaving the AAUAAA sequence on the pre-mRNA. An enzyme called poly A polymerase then adds a string of approximately 200 A residues, called the poly A tail. This modification further protects the pre-mRNA from degradation and is also the binding site for a protein necessary for exporting the processed mRNA to the cytoplasm. Pre-mRNA splicing eukaryotic genes are composed of exons, which correspond to protein coding sequences. Exon signifies that they are expressed, and intervening sequences called introns. Int-ron denotes their intervening role, which may be involved in gene regulation but are removed from the pre-mRNA during processing. Intron sequences in mRNA do not encode functional 15.4, RNA processing in eukaryotes 381 proteins. The discovery of introns came as a surprise to researchers in the 1970s who expected that pre-mRNAs would specify protein sequences without further processing, as they had observed in prokaryotes. The genes of higher eukaryotes very often contain one or more introns. These regions may correspond to regulatory sequences. However, the biological significance of having many introns or having very long introns in a gene is unclear. It is possible that introns slow down gene expression because it takes longer to transcribe pre-mRNAs with lots of introns. Alternatively, introns may be non-functional sequence remnants left over from the fusion of ancient genes throughout the course of evolution. This is supported by the fact that separate exons often encode separate protein subunits or domains. For the most part, the sequences of introns can be mutated without ultimately affecting the protein product. All of a pre-mRNA's introns must be completely and precisely removed before protein synthesis. If the process errs by even a single nucleotide, the reading frame of the rejoined exons would shift, and the resulting protein would be dysfunctional. The process of removing introns and reconnecting exons is called splicing, figure 15.13. Introns are removed and degraded while the pre-mRNA is still in the nucleus. Splicing occurs by a sequence-specific mechanism that ensures introns will be removed and exons rejoined with the accuracy and precision of a single nucleotide. Although the intron itself is non-coding, the beginning and end of each intron is marked with specific nucleotides, GU at the 5' end and AG at the 3' end of the intron. The splicing of pre-mRNAs is conducted by complexes of proteins and RNA molecules called spliceosomes. Visual connection figure 15.13. Pre-mRNA splicing involves the precise removal of introns from the primary RNA transcript. The splicing process is catalyzed by protein complexes called spliceosomes that are composed of proteins and RNA molecules called small nuclear RNAs, snRNAs. Spliceosomes recognize sequences at the 5' and 3' end of the intron. Errors in splicing are implicated in cancers and other human diseases. What kinds of mutations might lead to splicing errors? Think of different possible outcomes if splicing errors occur. Note that more than 70 individual introns can be present, and each has to undergo the process of splicing, in addition to 5' capping and the addition of a poly A tail, just to generate a single, translatable mRNA molecule. Link to learning. See how introns are removed during RNA splicing at this website, http://openstax.org/l/RNA_splicing. 382 Chapter 15, Genes and Proteins access for free at openstax.org. Processing of tRNAs and rRNAs. The tRNAs and rRNAs are structural molecules that have roles in protein synthesis. However, these RNAs are not themselves translated. Pre-rRNAs are transcribed, processed, and assembled into ribosomes in the nucleolus. Pre-tRNAs are transcribed and processed in the nucleus and then released into the cytoplasm where they are linked to free amino acids for protein synthesis. Most of the tRNAs and rRNAs in eukaryotes and prokaryotes are first transcribed as a long precursor molecule that spans multiple rRNAs or tRNAs. Enzymes then cleave the precursors into subunits corresponding to each structural RNA. Some of the bases of pre-rRNAs are methylated, That is, a CH3-methyl functional group is added for stability. Pre-tRNA molecules also undergo methylation. As with pre-mRNAs, subunit excision occurs in eukaryotic pre-RNAs destined to become tRNAs or rRNAs. Mature rRNAs make up approximately 50% of each ribosome. Some of a ribosome's RNA molecules are purely structural, whereas others have catalytic or binding activities. Mature tRNAs take on a three-dimensional structure through local regions of base pairing stabilized by intramolecular hydrogen bonding. The tRNA folds to position the amino acid binding site at one end and the anticodon at the other end, figure 15.14. The anticodon is a 3-nucleotide sequence in a tRNA that interacts with an mRNA codon through complementary base pairing. Figure 15.14. This is a space-filling model of a tRNA molecule that adds the amino acid phenylalanine to a growing polypeptide chain. The anticodon OG binds the codon UUC on the mRNA. The amino acid phenylalanine is attached to the other end of the tRNA.